This is an AQA psychology revision video for the topic of synaptic transmission, which appears on the A-level specification under biopsychology and the biological approach, as well as the GCSE specification for the paper 2 topic of brain and neuropsychology. So a typical synapse you will see will look like this. It's made up of lots of different structures and lots of different processes which you will need to be able to understand and describe. So this is the sort of image that you might want to draw out into your notes to learn from. I'm just adding on some little forks and the meaning of those will become clear shortly. The image will change slightly as I talk through the process, but if you'd just like to pause the video and copy out a similar diagram so that you can, in a different colour pen, label on the structures and the processes. So I'm going to continue to use the image on screen briefly to talk through the process, and then as I say, the image will change as I talk through and emphasise the key processes and structures more clearly. So what you see in front of you are two neurons. The green one would be called a presynapse and the grey one would be called a postsynapse. And what happens in our brain is an electrical impulse, or A-level students would they would prefer you to call this an action potential. It's the same thing. It travels down the presynapse. Now that's in the form of an electrical current, so that will cause these orange dots, which are the neurotransmitters, inside the purple circles, which are vesicles. The action potential, or the electrical impulse, causes the neurotransmitters to be released out of their vesicles, and as you can see, they are over here in the synapse. So once they are in the synapse, they then act as a chemical transmission rather than an electrical. So the electrical one just pushes them out of their vesicles. The neurotransmitters then enter the synapse, but it's the chemical reaction of them binding onto those things that I added onto the diagram, these receptor sites. The neurotransmitters bind to the receptor sites and will cause the action potential, so this time the electrical impulse, to continue down the postsynapse. So that's sending a message from one synapse to another, and that process is ongoing in our brains constantly. So let's have a look at that in a bit more detail. So information gets passed down the axon of a neuron, which is the end part, and this long slender fibre, as you can see here, of the neuron, as the electrical impulse, or as I've said, A-level students should be calling that action potential. So the action potential reaches the end of the axon and it will cross this microscopic gap here, which is the synapse, which you saw in a bigger scale on the previous slide. In the axon terminal, that's where the vesicles hold the neurotransmitters, and that action potential, that electrical impulse, causes the neurotransmitters to be released from their vesicles. So now the image will change to show you that these neurotransmitters have been released from their vesicles and entered the synapse. They then bind to the receptor sites on that postsynaptic neuron or postsynaptic cell, which causes the receptor sites to become activated. And that is essentially the carrying of the message. However, for both GCSE and A-level, you do need to understand the concept of excitatory and inhibitory effects. So once the receptors are activated by the neurotransmitters will produce either excitatory or inhibitory effects on the next synapse, on the postsynapse. So what we mean by excitatory and inhibitory is there are different types of neurotransmitters in our brain. Inhibitory neurotransmitters typically calm down our brain and nervous system. So it's something that can help return the body to calm. What happens is, if the neurotransmitter is inhibitory and it binds to the receptors of the postsynapse, it doesn't stop the cell firing, because the cells are always firing in our brain, but it means that message is less likely to be passed on. That postsynaptic cell becomes less likely to fire. Then on the opposite end of the spectrum, you have the excitatory neurotransmitters, which stimulate activities in areas of the brain. So excitatory neurotransmitters will make the postsynaptic cell more likely to fire. They just carry more of a charge, if you like. Now, a nerve cell can receive both excitatory synaptic potentials and inhibitory, and it just basically means whichever one there is more of will win. So it's the process of summation. They get summed together, and if the net effect of the neuron is inhibitory, then it's less likely to fire. But if there's more excitatory, if the net effect is excitatory, then it will be more likely to fire. So it's just about understanding that neurons can be both inhibitory and excitatory neurotransmitters. So that is synaptic transmission in under 6 minutes. Thank you for watching this video. I hope you found it useful.